The retroviruses comprise a diverse range of animal viruses united by a unique mechanism of replication. Retroviral replication has two particularly remarkable features. First, it reverses the usual direction of information flow in biological systems, i.e., from DNA to RNA to protein. Retroviruses have RNA genomes, but when a retrovirus infects a host cell, the viral reverse transcriptase makes a DNA copy of the viral genome. The second unusual feature of retrovirus replication is that it involves the integration of the viral genome into the chromosomal DNA of the host, a process mediated by the viral enzyme integrase. The integrated DNA form of a retrovirus is referred to as a provirus. Virions are spherical and enveloped by a lipid bilayer derived from host cell membranes. Two glycoproteins are associated with the viral envelope. There's a transmembrane or TM glycoprotein that's anchored into the viral envelope, and then a surface glycoprotein or SU that's found on the exterior of the virion. The SU associates with TM through non-covalent interactions. Under the viral envelope, the matrix protein, or MA, associates with the cytoplasmic tails of TM, as well as with the lipid envelope. They are genetically diploid with two copies of a positive sense single-stranded RNA genome packaged into the virion. Carried within the capsid are a few molecules of the enzymes reverse transcriptase, or RT, integrase, or RIN, and protease, PR. These virally encoded enzymes are required for the initial steps in the replication of the retrovirus. Two molecules of transfer RNA are also found within the virion. They are associated with the RT and are base paired to the genomic RNA near its 5' end at a sequence called the primer binding site, or PBS. A molecule of tRNA is the primer for DNA synthesis, and so the virion, before it buds from a host cell, acquires this tRNA from an infected host cell and carries it with it. The genomic RNA is a single-stranded molecule of positive polarity. A single molecule of cellular tRNA, which has a role in initiating reverse transcription, is attached to a primer binding site, the PBS, located a few hundred bases from the 5' end of the RNA genome. Also indicated on this figure are sequences important for replication, the PBS, or primer binding site, and PPT, the polypurine tract. There are four essential coding domains, GAG, PRO, POL, and ONV, that are found in the same relative positions in all replication-competent retroviruses. The GAG domain encodes the structural components of the viral core, the capsid, matrix, and nucleocapsid proteins. The PRO domain encodes the viral protease, while POL encodes the reverse transcriptase and the integrase enzymes. The ONV domain encodes the surface, or SU, protein and the transmembrane, or TM, glycoprotein. These two glycoproteins are responsible for viral attachment and penetration. Of course, the initial step in infection is attachment, in which the retrovirus utilizes one of its glycoproteins to attach to one or more particular cell surface receptors on the host cell. Some retroviruses also utilize an optional receptor referred to as the co-receptor. The second and third steps are penetration and uncoating. Retroviruses infiltrate the host cell by direct fusion of the virion envelope with the plasma membrane of the host. So let's look at HIV as a specific example as to how attachment occurs in association with co-receptors. We'll talk about HIV because it's probably the best studied of all the retroviruses. So once HIV comes into contact with the T cell, it has to attach itself to the cell so that it can fuse with the host cell and inject its genetic material. Attachment is a specific binding between proteins on the surface of the virus and proteins that serve as receptors on the surface of the T cell. Normally, these receptors help the cell communicate with other cells. Two receptors in particular, CD4 and a beta-chymokine receptor, either CCR5 or CXCR4, are used by HIV to latch onto the cell. On the surface of the viral envelope, two sets of proteins, also known as antireceptors, called GP120 and GP41, or SU and TM, attach to CD4 and CCR5 or CXCR4. After attachment is completed, viral penetration occurs. Penetration allows the nucleocapsid, the genetic core of the virus, to be injected directly into the cell's cytoplasm. GP120 actually contains 3 sugar-coated glycoproteins, and once GP120 attaches itself to CD4, these 3 proteins spread apart. This allows the GP41 protein, which is normally hidden by the GP120 proteins, to become exposed and bind to the cytokine receptor. Once this has occurred, the viral envelope and the cell membrane are brought into direct contact and essentially fuse into each other. Once HIV has penetrated the cell membrane, it is ready to release its genetic information into the cell. The viral RNA is protected in the nucleocapsid. The nucleocapsid needs to be partially dissolved so that the virus's RNA can be converted to DNA, a necessary step if HIV's genetic material is to be incorporated into the cell's genetic core. Okay, so here's another illustration of this. You can see here that GP120 binds with CD4. That leads to a conformational change in GP120 so that you get binding to the chemokine co-receptor. This leads to further conformational change in GP120 that exposes the fusion peptide of GP41, which then permits fusion of the viral and cell membranes so that the virus can penetrate the cell. Recent research has shown that most infecting strains of HIV use the CCR5 co-receptor in addition to the CD4 molecule to enter the target cells. HIV-infected people with a specific mutation in one of their 2 copies of the gene for this receptor may have a slower disease course than people with 2 normal copies of the gene. Rare individuals with 2 mutant copies of the CCR5 gene appear, in most cases, to be completely protected from HIV infection. Mutations in the gene for the other HIV co-receptors also may influence the rate of disease progression. So, of course, this has led to all sorts of research in, looking for drugs to control HIV. And in fact, a whole category of drugs called attachment or entry inhibitors are being studied in clinical trials. These drugs block the interaction between the cellular receptors and the anti-receptor on the virus by binding to or altering the receptor sites. Drugs called fusion inhibitors prevent the binding of GP41 and the chemokine receptor. So again, by knowing the molecular replication of a virus, that will enable you to potentially identify and develop drugs to treat and control the virus. Okay, now let's look at reverse transcription. It's kind of complicated, but we'll get through it. This figure shows in cartoon form the steps that are involved in the conversion of the single-stranded RNA genome found in virions into double-stranded DNA. In the figure, RNA is shown in pink and DNA in blue. For simplicity, the 5' cap and the poly A tail, which are present on the viral genomic RNA, have been omitted. So take a second to look at that input RNA up at the top of this figure for a second. Remember those terminal repeat segments, the R's, at each end of the RNA transcript. Those are going to be important now, and the U5 and the U3. So remember, we have a tRNA attached at the primer binding site, or PBS. The tRNA is going to act as a primer for the transcription of DNA from the RNA. The reverse transcriptase, of course, is going to carry out this process. What's going to happen is that a minus strand is going to be synthesized from the RNA template. But at the same time as this occurs, you're going to notice that the RNA template starts to disappear. That's from the RNase H activity of the reverse transcriptase. When you have an RNA DNA duplex, the RNase H activity degrades the RNA that's been copied. So where you've transcribed DNA, in this case, you've lost that RNA that acted as the template. So you're making minus strand DNA. Think about the direction that you have to go for transcribing DNA. You're going to find that you're running out of template. You come to the end of the RNA template. So you have to make a jump to the other end. And that's where the terminal repeat segments become important, because you've got identical R's at either end of the RNA. So when you've made that complementary minus strand DNA R sequence, it can bind with the R sequence at the other end of the template. So you get a jump to the 3' end of the genome. Those R sequences anneal, and transcription continues to the 3' end. And again, as this goes along, the RNA template is being degraded. Okay, so let's continue on. Remember the PPTs, the polypurine tracks that are immediately adjacent to the U3. Well, it turns out that the PPTs are relatively resistant to RNase H. So they're not digested when the rest of the RNA template is. And they're going to serve as primers for plus-strand DNA synthesis, because you're going to need to make a double-stranded DNA template in order to have something that you can integrate into the host chromosome. So you can't just make a single-stranded DNA transcript of the RNA. You also have to make the positive strand DNA as well. So you've made the jump. You've made a full-length cDNA copy now, and we have the PP segment, the polypurine tract. That's the only bit of RNA from that original input that remains. That is going to act now as the primer so that you can transcribe positive sense DNA from the negative sense DNA. And again, remember the direction of transcription when you're making DNA. So you're going to run out of template again. So now you're going to have a second translocation. In this case, you've got U3R, U5 PBS that have been transcribed into a positive sense strand. The molecule can circularize, and the two PBS sequences can bind together, and transcription will continue around the rest of the minus strand template. But notice now, you've made a double-stranded DNA as your final result, but it's not identical to the RNA template that came in. Look at the two ends of the DNA compared to the two ends of the original input RNA genome. You can see they're different. So we have what are called LTRs at each end. The DNA transcript is not identical. So let's look at this in a little bit more detail. Okay, so in this illustration you can see the RNA genome, the input RNA genome at the top, and then at the bottom the provirus. This is going to be the DNA double-stranded transcript of that original RNA genome. So reverse transcription is the conversion of a single-stranded RNA copy of the genome into a double-stranded DNA. To avoid the loss of genetic information, the double-stranded copy is longer on both ends than the single-stranded RNA from which it's derived. Although the genomic RNA has both a 5' cap and a poly A tail, what is most important is that the ends of the genomic RNA each have short duplications called R. After reverse transcription, the DNA copy is flanked by longer duplications called the long terminal repeats, or LTRs. The LTRs have in them the sequences such as enhancers, promoters, polyadenylation signals, etc., that are needed for the host machinery to generate the genomic RNA from the reverse transcribed DNA copy. The viral enzyme reverse transcriptase, or RT, is essential for the replication of a retrovirus. The RT is multifunctional with both RNA-dependent and DNA-dependent polymerase activity, as well as ribonuclease H, or RNase H, activity that degrades the RNA component of the RNA-DNA hybrid's duplex intermediate that's formed during reverse transcription. All of these RT activities are essential for transformation of the viral single-strand genomic RNA into double-strand DNA that can then be integrated into the host cell genome. The provirus, still associated with some viral protein, is transported to the nucleus as a pre-integration complex. The proteins of the pre-integration complex include integrase, bound to each end of the DNA, reverse transcriptase, and matrix protein. The pre-integration complex of some retroviruses, including HIV, is transported towards the nucleus using the microtubule network. The integrase cuts the DNA of a cell chromosome and inserts the provirus into the gap. Integration is completed by the DNA repair machinery of the cell. The integrated provirus genes may be expressed immediately, or there may be little or no expression of viral genes, in which case a latent infection has been initiated. If a latently infected cell divides, the provirus is copied along with the cell genome, and each of the daughter cells has a copy of the provirus. In the retroviral life cycle, proviral DNA is a template for transcription. Retroviral transcription is mediated by the host cell RNA polymerase II. The provirus is permanently integrated into the host cell chromosome and acts like a regular cellular gene, that is, it's expressed by the host cell transcription, RNA processing, and translation machinery. Upon transcription, the retroviral pre-messenger RNA is spliced into viral messenger RNAs that exhibit all characteristics of cellular messenger RNAs. That is, they all bear a 5' cap structure and a 3' poly A tail. In the case of HIV-1, for instance, alternate splicing gives rise to over 30 different messenger RNA species that are then exported to the cytoplasm by different pathways. HIV-1 messenger RNAs have been grouped into 3 classes according to their degree of splicing. So first we have full-length transcripts, which correspond to messenger RNAs that have not undergone any splicing process. These encode for the GAG and GAG-POL polyproteins, or they can be packaged into progeny virions. Then there's a whole group of singly spliced transcripts, which generate the viral proteins ON, VIF, and VPU. And then lastly, we have fully spliced transcripts, which express REV, TAT, VPR, and NEF. Retroviral protein synthesis relies exclusively on the host cell translational machinery for ribosomes, tRNAs, amino acids, and all required initiation, elongation, and termination factors. So here, just as a little summary, remember the GAG-PRO-POL genes are translated from full-length messenger RNAs. And also the full-length unspliced RNA is what's packaged into the progeny virions. Spliced RNAs serve as messenger RNA for envelope and any of the accessory genes. The number of accessory genes and the patterns of splicing and so on varies from retrovirus to retrovirus, and we won't be concerned with that. Just realize that all of the messenger RNA originates from transcription of the provirus in the host chromosome and then is packaged into various kinds of different sizes, different types of messenger RNA by post-transcriptional modifications such as splicing. Viral exit follows these steps. First, you have transcription by RNA polymerase II, splicing and nuclear export of viral RNA, translation of viral proteins, GAG assembly and RNA packaging, budding from the cell membrane and release from the cell surface and virus maturation. 2 forms of budding can occur. In the EMs at the left bottom, A shows you budding for HIV-1. With this virus, you have assembly and budding at the membrane showing both immature and mature forms. Mouse mammary tumor virus is shown at B, and these particles assemble in the cytoplasm, followed by budding. So with this virus, we see the more normal, imagine me making air quotes, type of budding, where the capsid is fully assembled before it buds through the plasma membrane. And here's a review of the entire life cycle for the retroviruses. So you got it all in one place. When we look at the classification of retroviruses, we can see that there's a number of different genera in this group with a large number of different viruses. When we look at the alpha retrovirus, this is a large collection of avian leukosis and sarcoma viruses. ALS is commonly present in chicken flocks around the world. Exogenous virus is transmitted as an infectious agent. Major illness caused by this virus is a wasting disease characterized by anemia, immunosuppression, and poor growth. Endogenous viruses tend not to be expressed. When we look at the beta retrovirus group, we have mouse mammary tumor, which is an interesting virus. A lot of studies have been done because the promoters in the LTR respond to estrogen stimulation. With the gamma retrovirus, we have feline leukemia virus as a member. This causes T-cell lymphomas and immunodeficiencies in cats, and we'll talk more about that for the last two slides. Then we have PTLV, or primate T lymphotropic virus, which causes adult T-cell leukemia. These have a long latent period and can be transmitted by sex, breast milk, transfusion, and IV drugs. PTLVs infect a number of non-human primates in addition to humans. In any area, the strains of viruses isolated from infected humans are more closely related to viruses isolated from monkeys in the region than they are to human strains isolated from another area. Thus, the viruses seem to have been passed back and forth between monkeys and people. Then with the lentiviruses, these are the complex retroviruses like HIV, whose core is cone-shaped in the mature virion. All lentiviruses have a tropism for macrophages, which comprise the major reservoir of infected cells in an animal. Other cells can also be infected, and this can be important in the disease process. These establish a lifelong chronic infection which elicits a vigorous immune response that is unable to clear the infection. Most known lentiviruses cause severe disease in their native host after a long latent period. However, SIV, or simian immunodeficiency virus, produces an asymptomatic lifelong infection in its natural host, African monkeys. SIV causes AIDS when transmitted to Asian monkeys. Although HIV-1 and 2 both cause AIDS in people, HIV-2 is not as serious a pathogen. The latent period is longer, the virus is not as easily transmissible, and it is not spread as extensively as has HIV-1. HIV-1 is responsible for the vast majority of AIDS cases in people. With FIV, feline immunodeficiency virus, these infected cats may appear normal for years. However, infection eventually leads to a state of immune deficiency that hinders the cat's ability to protect itself against other infections. The same bacteria, viruses, protozoa, and fungi that may be found in the everyday environment, where they usually do not affect healthy animals, can cause severe illness in those with weakened immune systems. These secondary infections are responsible for many of the diseases associated with FIV. There is a vaccine, but it does make the cat test positive in future tests. Spumavirus is a genus of non-oncogenic retroviruses which establish persistent infections in many animal species but are considered non-pathogenic. Its organisms have been isolated from primates, including man, cattle, cats, hamsters, and so on. Spumaviruses have a foamy or lace-like appearance in cell culture and are often accompanied by syncytium formation. Feline leukemia virus is one of the most common infectious diseases in cats, affecting between 2 and 3% of all cats in the United States. Cats persistently infected with feline leukemia virus serve as sources of infection for other cats. The virus is shed in saliva, nasal secretions, urine, feces, and milk of infected cats. Cat-to-cat transfer of the virus may occur from a bite wound, during mutual grooming, and rarely through the shared use of litter boxes and feeding dishes. Transmission can also take place from an infected mother cat to her kittens, either before they're born or while they're nursing. Feluke does not survive long outside of a cat's body, probably less than a few hours under normal household conditions. Fluke adversely affects a cat's body in many ways. It's the most common cause of cancer in cats and may cause various blood disorders and can lead to a state of immune deficiency that hinders a cat's ability to protect itself against other infections. Because of this, just like with FIV, common bacteria, viruses, protozoa, and fungi that normally do not affect healthy cats can cause severe illness in Fluke-infected cats. And just like with FIV, again, these secondary infections are responsible for many of the diseases associated with Fluke. In approximately 10% of infections, a cat mounts an immune response that is sufficient to eliminate the virus from the bloodstream, but not robust enough to completely eliminate the Fluke DNA from the body. Cats with these regressive infections carry Fluke DNA in their bone marrow, and although they are not infective for other cats during this stage of infection, the virus may be reactivated at some time in the future, at which point they may begin to show clinical signs and become a source of infection for other cats. The only sure way to protect your cat from Fluke is to prevent their exposure to Fluke-infected cats. Keeping cats indoors away from potentially infected cats is recommended. If outdoor access is allowed, provide supervision or place cats in a secure enclosure to prevent wandering and fighting. All cats should be tested for Fluke prior to introducing them into a home, and infection-free cats should be housed separately from infected cats. Food and water bowls and litter boxes should not be shared between Fluke-infected cats and non-infected cats. Unfortunately, many Fluke-infected cats are not diagnosed until after they have lived with other cats. In such cases, all other cats in the household should be tested for Fluke. Ideally, infected and non-infected cats would then be separated to eliminate the potential for Fluke transmission. A relatively effective vaccine against Fluke is available, although it will not protect 100% of cats vaccinated, and it is not considered a core vaccine. Owners contemplating Fluke vaccination for their uninfected cats should consider the cat's risk of exposure to Fluke-infected cats and discuss the advantages and disadvantages of vaccination with their vet. Since not all vaccinated cats will be protected by vaccination, preventing exposure remains important even for vaccinated pets. Fluke vaccines will not cause false positive Fluke results on ELISA, IFA, or other tests. This is in contrast to what we saw with FIV. One of these tests that can be used is called an enzyme-linked immunosorbent assay, or an ELISA, and it's usually performed first as a screening tool and can be run at the vet's office. ELISA-type tests detect the presence of free Fluke particles that are commonly found in the bloodstream during both the early and late stages of infection. The indirect immunofluorescence antibody assay, or IFA test, is usually sent out to a diagnostic laboratory after a positive ELISA test to confirm Fluke infection and determine whether the cat has reached the late stages of infection. IFA tests detect the presence of virus particles within white blood cells, usually an indication of a more advanced infection. The majority of cats that test positive by IFA remain infected for life. In some cases, isolating the whole virus or detecting DNA of the virus using a PCR test can also be used. This can detect whether or not the virus has infected the bone marrow. Again, you'll need to talk to your vet to see which particular tests might be appropriate. There is a SNAP feline triple test, which I've got illustrated here, which is one that's commonly done in the vet's office.